Then I'll start. Good morning, everybody. I'd like to first thank Stijn and Aaron, with whom I've been interacting the last couple of days, for organizing this meeting. Although I realize there's actually a bigger team behind them, but I've had very nice contact with them. I'm very happy to be here today and to welcome you in your home places. I'm at work, actually. Spend 50% of my time in the office, 50% at home. So today I would like to discuss a bit of the research we perform in my group about photoactivated chemotherapy, which is also, the acronym is PACT. And PACT is not very far from impact, which I realized when preparing these talks. So I'm going to try to explain how I've been wandering as a chemist by training into the world of biology in the last 10 years to try to make light-activated anti-cancer drugs. So the question I had basically now maybe 10 years ago, I would say, was whether the chemistry I knew, which was, let's say, photochemistry of ruthenium compounds, could be transformed into light-activated drugs, and this is not this should not be taken for granted. So that's the chemistry I'm talking about. If you take molecules which contain ruthenium, ruthenium in the classification is in the same column as iron, so it can be seen as a big iron atom. If you take a molecule that contains this metal center and also ligands which have, let's say, large aromatic cycle connected and conjugated with each other, and you shine light on them, you have something very interesting that happens, a very selective photoreaction. And many people think of photochemistry as like a bomb, that a lot of radical processes happen, etc. But that's not at all what happens here. I have a very smooth and clean reaction where one bond between the metal and...
This is a test.
Okay. I have a fire test in the building. That's quite interesting. So when you shine light here, you break a bond between the metal and the sulfur ligand. This is very selective. So your sulfur ligand gets free, and it is replaced on the metal center by a water molecule, which can bind to heavy metal, but it's very weak binding. When you do that, the color of the solution changes, which you can measure by UV spectroscopy. For example, you see here the change of the spectrum initially of the ruthenium sulfur species that goes down here, and a new band appear that is typical for this ruthenium aqua complex. So this reaction was known. You can do this kind of reaction with blue light or green light, even sometimes works with red light. And what I at some point considered is whether this change of the chemical formula of the molecules could be used to trigger a drug, so to activate it, if it would be possible to shine light into the body. And so this is how a little bit the kind of a perfect case would look like on paper. You would have a patient with a tumor, for example, here in the lungs, in purple. You introduce a drug which is not doing anything, actually, in the dark. So the drug is going to distribute in the whole body, like most drugs, even the best targeted drugs nowadays. They still go in a lot of places where you don't want to have them. However, for a real drug which is already activated without light, all the let's say the orange halo here would do something because the drug is activated. But if your drug is not doing anything before light activation, then you don't really care because it's not toxic, it will not have biological effect. Still, you do want to cure the disease, so then the idea is you bring a lamp or laser, you shine light locally on the tumor. For example, in the lungs, you can put a light source quite easily in the lungs. And then at the place where you shine light, you activate the drug. So it becomes highly cytotoxic, but only at that place, not in the rest of the body. And if this is working properly, then you remove the tumor. At some point, the body is also going to remove your drug, because that's what the body is doing with almost all drugs. The drugs are evacuated. So if this works fine, you have cured the patient with minimal side effects here. The idea is really that the drug that is not in the tumor, that is in the other organs, doesn't do anything because it has not been light activated. So as a chemist, you could say, well, okay, we'll make molecules and we'll study their photochemistry and we'll pretend we are going to do that in human, but we'd actually never do it. And there are many people who do that because it's very difficult to get into humans, and it's not easier for me. So the pathway to, well, from the chemistry, let's say what you do in your chemistry lab as a chemist, and an actual drug that really cure cancer is very long and very wavy. So I've here kind of tried to identify a few points, a few key steps you have to do to get to humans. So the first thing, once your chemistry is working, you have to put it in a cell, and maybe for biologists it's not so hard, but for chemists it can be a step. That's a step we went through now 8 or 9 years ago. Then from a 2D cell culture, you can go in a 3D environment. This is also a jump that you have to do. And then at some point, well, already at this level, but you have to really ask yourself which disease you are targeting. And this is actually a complicated question. Cancer is not one disease. There are at least, well, there are more than 200 forms of cancer that are identified, many different grades for every organs. So you cannot say you propose a drug for all cancer. You always have to say which cancer, which organ, which stage, etc. Once you have defined that, you have to go to animals, and this is also very big steps. Does it work in an animal is a completely different question from whether it works in your lab in water solution. And then start something which is, well, somewhat related to science, but not something that scientists typically do. Is there a market for your drug? Do you have competitions with other forms to cure cancer? And if all these answers are positive, well, will you ever find an investor who wants to pay for the clinical studies? And we are not there yet, but we are certainly looking for some people. So if you know anyone interested in investing in what I will present you today, please let them know, contact me. And that's only if we go through this whole path, which is difficult and it's long, that we might go for more impact by showing that it's working in humans. So now people have gone through this path with other molecules in the past, and I wanted you to realize that light-activated drug is not just a fuzzy concept for chemists, but it's really going on in the clinics. This is a picture from the US, which was given to me by Professor Teresa Bush, Pennsylvania, and I think it really represents very well what's going on. So you have a patient here which was operated from brain cancer. The tumor has been removed. There's a cavity here, and the surgeons want to clean out the cavity by killing the last remaining cancer cells. So they have a light-activated drug over there. There's an optical fiber which goes onto a table which is next there, and which has a laser on it, very powerful, that brings the laser light into the patient. You see the robot above, which has been used for operating the patient. And what you see also in the back is that while the molecule is being activated, the doctors just have to wait. So you're adding, let's say, in your regular cancer removal procedure, one step where you activate the drug for 15, 20, 30 minutes, depending on what you need. So the things that are used in the clinics already nowadays, or some of them are already approved, some of them are being tested in the clinics, are called sensitizers. So PDT goes for photodynamic therapy, and PDT works by, so the molecule, the sensitizer molecule absorbs light and becomes excited. That's what the star is showing here. And it will transfer its energy to an oxygen molecule, which is nearby in the tissue. We have oxygen everywhere. And oxygen has also an excited state. So you basically put the light energy into oxygen. So this is really an oxygen activation mechanism, but you can't directly activate oxygen by just shining light on it. You need the sensitizer. And once you have your oxygen excited state, it's going to oxidize everything, your membranes of the cancer cells, the DNA, the proteins. So we'll break a lot of things, which will make cancer cell death. So you can see here, this is PDT applied in a esophagus cancer. Before PDT, you see the tumor here, which has a slightly different color. It's obstructing the esophagus. Right after PDT, the PDT, you see the action. It's doing tissue necrosis. It's been killing the cancer very actively. So here, the immune system is actually cleaning out all the dead cells, which is the reason why the tissue is a bit brown. And after some time, when the immune system has done its action, then your esophagus is again very healthy and looking very nice. So I'm going to show you alternatives to PDT, but I don't want you to go out of this lecture with the feeling that PDT doesn't work. PDT does an amazing job in many cases, and people have, because of that, addressed the question of shining light into organs by many different ways. Maybe the easiest way is for skin tumor. If you have a tumor on the skin, it's easy to just bring a lamp on the skin and shine light. But if you are in the body, I've shown you this kind of setups for brain tumor, where there is really kind of balloon which is going to scatter light onto the whole cavity where the tumor used to be to kill the cancer cell on the surface. This is a picture from the Dutch Cancer Institute, where there was a tongue tumor in a patient. And here they put a lot of optical fiber. You see here the kind of optical fiber people use to bring light into the patient. At the end of the fiber, there's a light diffuser. The light diffuser is put into a catheter, and it's then inserted directly into the tumor, and you get a lot of light. And here's an example for pancreas tumor, for example. Yeah, so there are many techniques to shine light. Now, we started, let's say, with cells grown in a petri dish, come back from the clinics, because we are developing new molecules. And I'm going to show you a lot of curves that demonstrate whether a compound is activated or not by light. So I wanted you to be introduced to what I'm going to show you later. So typically we have two plates with cancer cells grown in there. We want to test here 3 different drugs. And one of the plates is going to stay in the dark. The other one is going to be irradiated with light. So here we have 3 drugs in the same plate. We have also a control well. We have the same cancer cells, but no drug. And then we do what's called the dose response curve. So we count the cell at the end of the experiment. And when there is very little or no drug, you have 100% cell survival compared to the control. And when you put more and more of your drug, you kill your cell little by little. Now, if you do that, you can also shine light on your cells in presence of the drug. Then you will have a different dose response curve. Basically, for the dark plate and for the light plate, you can define a concentration for which you kill 50% of the cell compared to control. So this is called an efficient concentration, EC50 in the dark. You have also an EC50 in the light. And if your molecule is activated by light, then the curve is going to shift to the left, which means the EC50 is going to go down. You need less of your molecule to kill the same 50% of the cells. And the ratio of your EC50 in the dark by the EC50 in the light is called the photo index. So the photo index is a measure in vitro of how many times your drug is activated when you put it onto a cancer cell. Now, as I said, PDT sometimes work very, very well and sometimes doesn't work so well. So this is a compilation by Michel Heger, who is a PDT specialist. And for example, if you take skin tumors, you have 75% of the skin tumor that respond well to PDT. We have 25 that do not. And if you go to other forms of cancer, like biliary cancer, it's really not efficient at all. Now, I wanted also to mention that for Teresa Bush, which I already mentioned, she published a study about tongue and mouth tumors, where they looked at, let's say, the amount, well, the response to PDT for different patients that had these kind of tumors. And before doing the PDT, they measured the amount of oxygen, the pressure of oxygen in the tissue. And you see that the people who have a low oxygen concentration in the tissue, they usually have a very bad response to the PDT, while the people who have enough oxygen in the tissue, they have a good response to PDT, basically, because if there is not enough oxygen, it's difficult to have a PDT effect. Now, this problem of which is called hypoxia is a more general problem. It's not only for PDT. So this is a study about a uterine cervix cancer patient who, when they arrived for being treated, also were measured the median pressure of oxygen in the tumor tissue, and they identified a threshold around 10 millimeter mercury pressure of oxygen into the tumor. And if the people had more oxygen than this threshold, they had a better, let's say, life outcome. And we are looking here at the overall survival probability for all treatments together, so radiation, surgery, chemotherapy, etc. While if you have little oxygen in the tumor tissue, then you have overall more chance of dying. So the problem of hypoxia in oncology is a serious problem, not only for PDT, where it's kind of obvious, but also in general. It's a good problem to tackle because there is a clinical need there. In vitro, what we measured a long time ago, the basic idea is that when you are far away from the tumor vasculature, well, the cells that are, let's say, far enough, they still have a little bit of oxygen, so that they still live. They will be dormant. The ones that are too far, they die because they don't have any oxygen. But the ones that are close enough but far enough at the same time, they're hypoxic. And they have very little oxygen, but they are still cancers, still living. So they are the really bad guys. And the cells which are close to the tumor vasculature, they have a lot of oxygen. They are easier to kill because they are well, let's say, distributed. They have a good blood distribution. So the chemo and the PDT effect, if you do PDT, would be very efficient. Now, in vitro, as I said, you can measure the dose response curve here of lung cancer cells. So in the dark, we see no toxicity. In the light, using rose bengal, which is a very well-known PDT molecule, you see good killing at, let's say, 1 micromolar here. When you go in a hypoxic situation, there's only 1% of oxygen on the cells, then you see that your PDT effect is kind of quenched. So this is what we are trying to address. And we address them with ruthenium molecules, as I said. And the idea is the following. We are going to remove oxygen from the whole picture of the photoreactivity of a light-activated molecules. So that's what PACT means. It's a kind of chemotherapy molecule, but instead of being active straight away, you need light to activate it, but you don't need oxygen. That's quite critical. So the idea is that your prodrug is actually made of two parts. One that contains the ruthenium metal and one that does not, which we call the ligand, L. And we're going to start with a case where when you shine light on this, you break these ruthenium ligand bonds and you free your ruthenium molecule, which is cytotoxic. So one typical way for heavy metal compounds and drugs to be cytotoxic is to bind to DNA. And the cancer cells don't like this. And the idea is if you do that a bit too much, you're going to tell the cell the DNA is damaged and it has to go into apoptosis. So the whole idea of this PACT concept, at least that's how we developed it at the beginning, was really to say this mechanism of action, this breakage of a molecule in two pieces, is a priori independent from oxygen. I should also say it's potentially a bit more selective than PDT, where you simply generate ROS. So here we don't generate per se ROS. If your ligand or if your ruthenium activated molecule are very selective, for example, the target a protein which is overexpressed in cancer, then you would have something which is much more selective than PDT as well. We're trying to, in general, solve the problem of PDT as well. So maybe develop molecule that will provoke less photosensitivity for the patient, provoke less tissue necrosis, and if possible, that are active when other treatments fail. Now, a little bit of photochemistry, because this is where my background lies. So when you take a ruthenium molecule, you shine light and you get what is called an excited state. And photochemistry can be quite tricky. Sometimes from this excited state, you get nothing. So nothing happens, the energy comes down, and you have no chemistry at all. You lose your energy. In other cases, you have phosphorescence. This is used for some things called PDD, so photodynamic diagnostic. It can be very useful for detecting a tumor. What I've discussed up to now is a form of photoreactivity which is involved energy transfer. It's called PDT, type 2, for people who are in the field. As I said, you put the energy of the ruthenium onto the oxygen molecule. You can also do electron transfer from the ruthenium to oxygen, and you will generate ROS. I haven't discussed too much of that. I'm not going to dive into this because I want to focus on the photosubstitution. And photosubstitution is a reaction where a ligand, as I said, is substituted by another one. So this is how it's working. You have your ground state molecule. You will see the ruthenium in blue, the sulfur atom here bound to it, and the ligands in gray and blue over there. And these ligands make that there is a pi star orbital, which is on these ligands. And the electrons that we're going to excite by light are the electrons that are based on the metal. So these T2G and EG orbitals are centered on the metal. So when you shine light, you see you take one of these electrons, you put it over there. Well, you will have a spin flip as well. I don't necessarily have to go too much into detail here. So this is the kind of excited state that is capable. It's called an MLCT, metal to ligand charge transfer. It's an excited state that is capable of transferring energy to oxygen, for example. So for doing PDT, you will use these kind of excited state. And in the molecules we are working with, there are other kind of excited states, which are called metal-centered. Because you see that this electron here, which has been excited by light, is going to be promoted to the orbital which are over there, which are called EG orbital, and they happen to be anti-bonding. So when you put an electron over there, you weaken some of the bonds between the metal and the ligand. So what you can see here by calculation, that the ruthenium-sulfur bond in this particular excited state has gone from, let's say, 2.4 angstrom to 3.1 angstrom. So there is a very significant elongation of the bonds. So if you do that in water, there's a lot of water molecules around. They will bump the sulfur ligand out because it's weakly bound to the metal, and you will get your photo product, which is the aqua complex. So now if you look at, let's say, a bit more energetically wise, let's say you have a ground state, you get your MLCT state and a triplet MLCT state. As I said, you can have phosphorescence or you can have energy transfer. So for the molecules that are good at doing PDT, from that excited state, which is called the MLCT, you will pump basically the energy of the regular oxygen molecule into the excited state oxygen molecules, which is called singlet oxygen. You pump that guy by losing the energy of the sensitized excited state. This is what you get when your metal-centered state, the dissociative states on ruthenium, are very high. Now, with the molecules we are working with, these states are much lower. So provided you have, let's say, at room temperature at 37 degrees, that's fine. You have enough thermal energy to get to these dissociative states to get your photosubstitution. But you should realize that when you do this, then it will occur at the cost of singlet oxygen generation. So the interesting point is that the molecules that are good at doing photosubstitution, they are very bad at doing singlet oxygen. So the PACT molecule are really intrinsically not good for doing PDT. So for example, if I take that molecule, and this is a very well-known molecule in photochemistry, it absorbs in the blue, so it's slightly yellow. And if you shine blue light on it, you get a nice orange emission. So this comes from the emission of that molecule. You have 2% quantum yield for emission. But actually, this molecule is very good at doing singlet oxygen. So 73% of the excited state will end up into making singlet oxygen. And it's very bad at doing photosubstitution. So none of the ligands are going to be kicked out when you shine light on this. Now look at the difference with the next molecule I'm going to show. I'm going to change that ligand over there. So I will transform one of the pyridine rings into a thioether. And when you do that, you completely change the photoreactivity of the molecule. You see that when you shine blue light, then you have a huge change of the color of the molecule, as we've seen before. And if you analyze your reagent, you will see that the NS ligand here is kicked out very completely, selectively, and you make a bis aqua molecule that might be able, for example, if this would happen in the nucleus, that would be possible to bind to DNA and kill a cell. So this molecule does photosubstitution, 0.3%. It is very bad at doing singlet oxygen, and not at all. It's a very bad photosensitizer, and it does not do any phosphorescence because all the excited states get down either via photosubstitution or by non-relative transfer. Now, still, this molecule is not a good PACT molecule. It's light activated in a chemical sense because this ligand is kicked out by light. However, if you put it onto cells, cancer cells, and you shine light on them, you see that your dose response curve basically stays desperately flat. So here's a blue light activation experiment, which means basically the molecules do not care whether this molecule is activated or not. So it's biologically inactive while it is chemically activated. The problem with this molecule is that it's too hydrophilic. The hydrophilicity of the molecule, so the water solubility, can be measured by something is called the log P. And negative value mean hydrophilic molecules, and positive value means hydrophobic molecules. So this molecule has a log P of -1.33, so it's quite water soluble. So it does not pass membranes. It will not go into the cancer cell. So you make your photoreaction outside the cell and nothing happens to the cancer cell. That doesn't work. Now, if you want to transform that molecule into a molecule that does work, you need to put more hydrophobicity. So that's quite easy to do as a chemist. We make new molecules. You see, we introduce more phenyl groups over there. So we improve the greasiness of our molecule. For the rest, it's the same. There's a ruthenium 2+ center. There is the same NS ligand as before. I mean, it looks quite similar, but now you see the log P is plus 0.29. So it's much more hydrophobic. It will cross the membranes. If you shine light on it, you get the same kind of transformation, same kind of color change. So we have all the characterization that the NS ligand is getting kicked away when you shine blue light. And this time, when you put it in cell, you see first that it's toxic in the dark already a little bit. Actually, it's quite toxic in the dark. 3 micromolar is similar to cisplatin. But when you shine blue light, you get much higher cytotoxicity, 0.5 micromolar. So the photo index here is 5.5. Yeah. So now we have demonstrated that, well, PACT seems to work this time because the molecule is capable of going into the cell and being where the photochemical activation does something. Still, let's say maybe I went too fast. Further, what I wanted to say is that the EC50 is a little bit too high, actually, because we would like you, well, the best would be to have a molecule that in the dark doesn't do anything. That's what I told you at the beginning. And obviously, this molecule is already very cytotoxic in the dark. And the problem is it comes into the cell in far too high extent. So as the other molecule was too little hydrophobic, this one is too much hydrophobic. It comes into the cell in too high amounts. So we went for a kind of intermediate case where we kept one of the small ligands we had in the first molecule. We kept one of the greasy ligands we had in the second molecule, and we kept our NS ligand, which is kicked away by light. So this is kind of a fine-tuning of the hydrophobicity. And now you see what it does in the cell, and so it's working well. We have a higher PI, and the thing is, the critical thing is that the phototoxicity in the dark is much lower now, so your EC50 is much higher, which means less cytotoxicity. If you compare it to the previous molecule, that's what I showed you before, so you shift both curves a bit to the right, but you improve the photoactivation. And so the critical point here is really that this molecule is less hydrophobic than this one. So here we tested how much ruthenium you get in the cell. So you calculate per milligram protein how much ruthenium you have. And if you take this molecule, you find 2 nanomole of ruthenium per milligram protein. But this is when we treated the cells at a concentration of 3 macromolar. While this one, to get the same amount of ruthenium in the cell, you need to treat it at a much higher concentration. So this shows really that you need to put much more of this molecule to have it entering the cell. So this one is a much better hydrophilic to hydrophobic balance, the top molecule over there, compared to this one, which is too hydrophobic, that black marked in that kind of yin and yang representation. So this is all, well, how the chemistry relates to the biology in vitro. Now, we've tested this in lung cancer cells. And at some point, I received a proof of concept grant from the European Research Council. And this is very important because this is really trying to fund specifically, let's say, experiments or things that you would need to go to come closer to the clinic, so to improve the impact. So there we had budget for making a market report, which was trying to see what is the highest market attractiveness of PACT, which kind of cancer within these 200 kind of cancer would be the most attractive, not scientifically speaking, not for the patient, but for on the point of view of the companies, on the pure, let's say, investment technological point of view. And so we asked the company to do that, and they looked at different things. So the epidemiology of different cancer. For some cancer, you have a lot of patients. For others, you have very little. So this obviously makes a huge difference in terms of return on investments, but also what is the current treatment option possible? Are there any unmet clinical needs? Competition with PDT? Is hypoxia playing an important role? And they also interviewed some oncologists who are operating patients, etc. And they came up to, let's say, 3 main cancers that were of, let's say, best technological use for company development, which were skin non-melanoma, so basal cell carcinoma also, head and neck cancer, tongue, mouth, and eye cancer, which I'm going to focus on in the rest of the talk. I should mention here that this is really purely seen on the basis of market interest. So it's not that we only focus on the 3 cancer. You will see some other forms of cancer because there is also other questions related to other patient needs or scientific interests. Now, this molecule, which you have seen just before, as I said, we started to test it in eye cancer because that was one of the interesting cancers from that report. We also happened to test it in prostate. I'm going to show you well, there will be two eye cancer cell lines, which are a bit different, but also quite similar. And we will compare it to prostate cancer because it's also quite relevant scientifically and in terms of for the patient. So now we measured the photo index. You see the molecule is working very well with a PR around 10. In prostate, actually, it's even higher. We have very, very good photo indexes over there. So more than 30 times more cytotoxic after green light irradiation than in the dark. And we started to work with Professor Sna Jagalska and her PhD student, Quanchi Chen. They are specialists of zebrafish, which is a first type of in vivo model. And zebrafish are very, very tiny. So they have been proposed as a way to test molecules in vivo because they are much smaller than mice, obviously. They're also transparent, so it's much easier to look into a zebrafish embryo. You can look into their vasculature. You see here the green color is used for looking at the veins. The tumor cells, they can be colored in red, and then you can really see where's the cancer, how big is it, and how does it evolve upon your treatment. The fact, obviously, that the animals are transparent is very nice for phototherapy because not only you can look into them, but you can also activate the drug everywhere in the body of the embryo just by shining light through it. Another nice point of the zebrafish, if you want to look at side effects, and we're trying to minimize the side effect, is that for zebrafish embryo, the whole, let's say, toxicity is very, very well characterized. And there's a lot of science on these model. So we started working with them. We of course, use their protocol. All the red will be the cancer cells, which have been, well, stained. Then the vasculature is in green, and we started to realize that testing in vivo is not enough. You have to know what kind of model, and this reflection on the model is very, very important to realize because all the results we're going to have in vivo really depend on the kind of model that you have. So as a chemist, if you don't, if you are not interested in these things, you might just test the wrong cancer model, and then you will have no impact just because you use the wrong model. So I'm going to show you about two types of model. The ectopic model is when the prostate cancer cells, for example, end up in the tail. So which means the tumor model does not end up in the place where it was supposed to be, so to say. While in an orthotopic model, if you take, for example, eye cancer, they would end up in the eye. So an eye cancer type in the eye tissue is much closer from the real situation of a real patient than when the tumor is not really in the place where it's supposed to be. So we have tested different types of giving these PST compounds to the embryo. So one is to simply put the compound in the water where the embryo swim. Another way is to inject the compound in the intravenously. And basically, the compound is going to start circulating in the animal. We use a dose to light interval of 1 hour. And we can also put the compound, inject it retroorbitally, which means just behind the eye, actually where you put the eye cancer cells as well. So we've tested these 3 ways of administration, and the idea is that 2 days after fertilization, you inject the cancer cells, which are red labels, so they will settle down and make the tumor. So at 3 days, they have made a tumor, and at 4 days, you will inject the compound, then shine light, and repeat the treatment. So we have repeated this treatment 4 days in a row, 60 minutes each for light activation. And we look at the tumor burden by microscopy, because we can directly look at the red and green emission. So you can see here, this is the case of a prostate cancer cell where we saw the PI was very, very high. And we treated at the maximum tolerated dose. And what you see here, you see the tumor in the dark without compounds. So that's a control, kind of. You can also shine light and you see nothing happens. So the light itself doesn't kill the tumor. Then we can also apply our compound and then not so much happen. And if we apply our compound and we shine light on it, well, actually not so much happen here. You can see these bars, basically they all stay more or less at the same level. So we saw no activity of our compound in that particular model. So this was an ectopic model, so where the prostate cancer cell end up in the tail, so they are not in the place where they're supposed to be. And basically the molecule doesn't work. Now we went for eye cancer using intravenous injection as well, and I will keep it short. You see that the effect was also non-measurable, so our compound was not doing anything when, let's say, the eye cancer was not in the eye, when it was settling down in the tail, and you would inject the compound in the circulation. However, if we would put eye cancer cell and inject them behind the eye, so retroorbitally injected, and then we would put the drug at the same place, then you see that we start to see a very interesting effect. Yeah? So for this particular cancer cell, you see that the PI is 8.5, which is a nice value, but it's also much lower than the one in the prostate cancer cell, which was more than 30. So that was a very important conclusion for me from these experiments, is that we are used to calculate the photoindex in vitro, and we usually say, well, the higher the PI is, the better it will be for in vivo activity. But actually, it's not the case. What really mattered here was really the way the tumor, well, the drug accessed the tumor. So it was really dependent on the type of model we were using. Which is, I think, very important as a chemist to realize that the in vitro to in vivo translation is kind of complicated. And you need to really understand what's going on in vivo if you want to have interesting results. Another thing is that obviously in a zebrafish embryo, it's difficult to activate the drug only at the place of the tumor because the animal is so small. Mouse are still more interesting to mimic, let's say, what's going on in a patient. But to go there, and also to finish my presentation, I wanted to jump to another compound. Up to now, I've told you that, well, your prodrug is kind of protected by the ligand, so it's the ruthenium part that had the toxicity. But we have realized we could also reverse the picture. We could consider a case where the ruthenium is actually non-toxic. I showed you the first compound I showed you. It was chemically activated, but once in a cell, it was not doing anything. And so there are indeed compounds like that that are not toxic by themselves. Huh? They are heavy metals, but they don't do anything. And on the other hand, there are also ligands which can be very cytotoxic. So the world of protein inhibitors for cancer treatment is very, very large. Many of these molecules are in the market. And the basic idea is that if your organic ligand binds to a protein which is very important for the tumor to grow and for the cancer cells to reproduce, to split, then you will kill the cancer cell by the action of your inhibitor. So, and then the idea is that you are going to use the non-toxic ruthenium compounds to cage this toxic ligand. So that would be the caged ligand. And after you shine light, you break the bond the same as before. But now this is the uncaged inhibitor. And your caging group, well, basically is also released, but it doesn't do too much. So we have investigated several compounds of these kinds, but I wanted to talk to you about the work we did in collaboration with Alexander Konyenko from Texas State University in the States. They were working on these kind of inhibitors. So there are a microtubule polymerization inhibitor. And as you can see, there are organic compounds that bind into the colchicine binding site of tubulin, and that prevent the tubulin to polymerize, which is very important for the cell to survive and to grow and to split. And so the idea, he had a compound with a thioether here. So we said, well, we like thioether because ruthenium binds to thioether very well. So the caging itself procedure, the synthesis is very easy to do. You take your aqua complex, you mix it in a solvent with the inhibitor, you heat a little bit, and then you do some chemistry, some purification, and you end up with your caged compound where the thioether is bound to the ruthenium. And you can see that you can imagine, I think, that if the ruthenium, you would have a huge ball here that would make it impossible for that particular inhibitor the colchicine binding site, because ruthenium is huge and would prevent that. Now, if you shine light on this complex, we have shined, for example, green and blue light, then you will break the ruthenium sulfur bond, as I discussed with you before. So you see the reaction is working very well. You see the color change over there. We've measured the quantum yield, so 0.5 or 0.6% with blue light, 0.4% with green lights. So this is behaving very, very well, as I told you before. Now, the question is whether this photoreaction will have any consequence on microtubule polymerization. So we have an assay here, which is a fluorescence assay. If you take tubulin and you put paclitaxel, which is an approved anti-cancer drug that forces the microtubule to block it, well, to be blocked in a polymerized state. You see a high fluorescence, so this is polymerized microtubule. Now, if you take the control, let's say, so the control is either no drug, nothing, then you have a certain polymerization rate, yeah? But you see that our compound in the dark does the same. So basically, our compound in the dark, or the control, where there is no ruthenium, it does exactly the same. And now if you take our caged inhibitor and you shine light on it, either green or blue, you see then you have these two curves, which means that basically the microtubule is blocked in a depolymerized fashion, which will also be bad for the cells. So we have worked also with Sylvia Ledevedek, who is a colleague from the Leiden Academic Center for Drug Research in Leiden. And she was able to image the microtubules in green. In actin, you see the red. So microtubule and actin are two sides of the cytoskeleton, which is very important for cells to live and to split. In blue are the nuclei. And what you see is that if we keep our compound in the dark, not so much happened to the microtubules. While if you put the compound here, 10 macromolar, 20 macromolar, you see all the green is kind of disappearing or strongly modified, while the red is still seems not to be touched too much. And so our compound is really under light activation, really acting on the microtubule, while if we take our vehicle control, nothing happens, obviously. So this is really a compound that under the action of light breaks down the microtubule polymers inside the cell, which in terms of toxicity, obviously, has dramatic consequence. So you see here the compound in the dark is over there. It has an EC50 of, let's say, well, 14 to 35, depending on the cell type. When you shine light, you get the green curve over there. So you get a strong enhancement of the toxicity, depending on the cancer cell line as well. So this is skin non-melanoma. You have 30 times more toxic compound. In lung cancer cells, it's a bit smaller. It's 4, but it's still quite clear. You see here is the free ligand and without any ruthenium, and the free ruthenium is not cytotoxic. So the free ruthenium is not cytotoxic, while the free ligand is cytotoxic. Now, as I told you, does it work on a hypoxia? So we did the same experiment, but growing cell in the at a very low concentration. And what you see here is that we keep low EC50 in the dark and high EC50 in the green light. In lung cancer cells, for example, you see that the EC50 is actually almost not changed. So this really is a demonstration that the light activation mechanism works obviously on normoxia, but it also works on hypoxia because this bone-breaking photoreaction does not require any oxygen to take place. What you will see, however, is that the value we get after light activation are quite higher here than what you had on a normoxia. And this is a problem that we are facing right now. These are dose response curve of two compounds from our group. So you see the dark and the green light curve. When you do it on a hypoxia, you see that both curves are basically switched to the right, which means both the dark toxicity becomes lower, but also the light toxicity becomes lower. And this is not, so the activation still works, but both EC50s becomes higher. And the problem with this is a biological problem. It's not a chemical problem. When a cell grows under hypoxia, you will trigger a lot of genes which allow the cell to survive to the low oxygen concentration. So the main gene is HIF1 alpha, but there are many others. So for example, you will overexpress efflux pumps, which are pumping out your drug from the cell, which will give more resistance, but there are overall, there are many mechanisms to resist to chemotherapy drugs under hypoxia. So in general, hypoxic cells are more resistant to chemotherapy than normoxic cell, and that's why all these EC50 go to the top. So we are working on this now. We are trying to really find molecules that are that remains as cytotoxic under hypoxia as they are under normoxia. And you can see here a picture showing you that these cells are really different. We look at different, let's say, proteins in a cell. So here's a skin non-melanoma cancer cell, and you see that if you look, for example, at cadherin, under normoxia or hypoxia, you have a completely different expression of that particular protein. For other proteins, it's the same, but for many proteins, it's strongly changed. So basically, hypoxic and normoxic cells are really different beasts to kill, and usually it's more difficult. Now, to finish with, we said we would like to really try working in mice, because there you can really shine light just on the tumor. So we went to colleagues in France who are used to doing PDT in mice, and they tested our compounds. This is very preliminary work, but in a nutshell, so this is lung cancer cells in a nude mice. What you can see is that the tumor growth in the dark or in the control is all these curves over there. And the only curves that stand out here and that shows 30% reduced tumor volume is the one that contains our compound that has been irradiated. So, of course, 30% is not yet great. We want to improve on this. But you realize from the zebrafish, we need to understand a lot of what's going on in the mice. And this was really the first test we did ever. We had no idea which concentration to use, which light treatment, etc., etc. So we have to work on this to understand better the distribution of the molecule in the mice and to improve on that number. So the conclusions for my talk, maybe for you to remember, the PACT, so photoactivated chemotherapy, this works by photosubstitution on a metal. So you break the bond between the ligand and the metal, and you generate two fragments, and you can either have one or the other that are more cytotoxic than in the dark. This reaction is independent from the presence of oxygen, so it is designed to work for hypoxia, but it will work in air as well. So we're not only doing things for hypoxic tumors. Either the ruthenium or the ligand can have an action on the cell. And I've also discussed with you a little bit that this problem of hypoxic cancer cells and hypoxic tumors is really significant in oncology. So we hope to work not only on a PDT, let's say, problem, but also in oncology in general. So before I finish, I've tried to put here a few of the important points which to me contribute to impact, because that's the theme of the day. I should say that I was greatly inspired by people, in particular my PhD supervisor, with whom I learned this photo substitution reaction in completely different context of molecular machines. Now, then you need a good research question, and I think I tried today to explain you our research question. Why is it interesting? Now, we need the right environment. Research environment is very important to have a lot of biologists to understand all the things you don't understand yourself, and with whom you can discuss. You need excellent people in a group that are both motivated and properly trained by you, but also by other people. So it's really important to have an excellent university for training all these people. Basically, you guys, because this conference is really about students, for students. Now, on my side, I realized that what is critical in my work is really to be able to discuss with biologists in particular, but I've also discussed a lot with physicists and theoretical chemists. Discussing with people outside your field is really critical if you want to improve the impact. Multidisciplinary research. As long as you only do one thing, just your chemistry, you can do awesome chemistry, but to reach impact, I think really needs to go outside, discuss with other people. In general, communication is quite important. You need money, and you need a little bit of luck to get money, for example. These are the awesome people I used to have in my group. There are only a few of them in my group now, so I really have to change that photo. But with the corona, there's nothing more difficult than making a group photo in the corona situation. I would like to mention Samantha Hopkins and Björka Sievert, two postdocs who have contributed to bringing biology in my group. It was really important. The chemistry I've shown to you were done mostly by Vincent von Rixel and Jordi Cuero-Garibo, who are now who have left the group. There are many bachelor and student and master students on this photo. We have many of them every year, so just come to us if you want to make an internship. And I think that's it for now. Thanks a lot for your attention. Just maybe a small mention of a photochemistry conference, which will take place in Amsterdam at the UvA in 2022. And also this year, for those who like photochemistry, we will have the Dutch Photochemistry Days on the 10th and 17th of May. If you are a PhD or postdoc, you can ask your supervisor to propose you for a talk, but everyone will be welcome to look at more photochemistry. Thank you very much. I'll be happy to answer the questions.
Yes, thank you very much, Professor. You can ask your questions in the chat, and I will forward them to Sylvestre. In the meantime, I have a question. What is the penetration depth of the light you use?
Yeah, so very good question. So it depends a lot on the color. So basically, blue and green light, they penetrate over a few hundreds, well, let's say 100 to 300 micrometer or so, which is not so much. For red light, it goes much further, so typically half a centimeter or so. But when you put several optical fiber in a tumor, you can. So if one fiber has an extension of, let's say, 0.5 centimeter, you can address relatively big tumors. And if you go to the near infrared, so 700 or 800 nanometer, then you really go 1 centimeter penetration depth. So this is what we are talking about. Yeah.
Okay, thank you. I don't see any questions from the audience yet. If you have one, please type it out. Okay, I don't think there are questions yet.
That's okay. I will be here in the networking event.
I found there's a question.
Yeah.
So when using green or blue light, you would insert some type of acupuncture needles that emit the light?
That's a possibility. There are people who actually generate small capsules that generate light inside the body. I've seen that in a recent conference. I should also say there is currently a clinical study for the use of PDT for curing bladder cancer, where you basically put the anti-cancer compound in the bladder, and the bladder cancer is at the surface of the bladder, and it's a very thin tumor type. And 20 years ago, people had done red light PDT for bladder cancer, and they had realized that there was some toxicity for the bladder muscle that were below the tumor tissue. So now they're trying with green light, precisely because it has a lower penetration depth, because you really want to kill the tumor tissue without harming the muscles that are below, let's say. So the lower penetration depth can be actually an advantage for phototherapy as well. So when I mentioned that every type of cancer was different, and you cannot cure one cancer with one molecule, one type of light, but you really have to focus completely on which cancer, which grades, which kind of light, etc. It's very, very precise. This is what we are talking about. So if you have a big tumor, you would like to go for red or infrared light. But if you have a very thin tumor on the skin or in the bladder, you would prefer some light that does not penetrate too much.
Okay. Now some questions have come in. Firstly, is it possible to change the ligand depending on the type of cancer?
Yes, completely. So basically, I've shown you a couple of ligands which we have worked with. But if you look into a database of all the approved drugs, there are many, many drugs that have either nitrogen ligands or sulfur ligands, or even oxygen ligands, and that we could use as well. So for normal chemotherapy, you also don't get any chemotherapy agent if you have a cancer. And so the oncologists really go for your particular kind of cancer. They now even look at the mutation of your personal cancer. That is called personal medicine, and then they're looking at that in huge databases, which drug is more efficient for that particular form of mutations. So definitively, this is something that we could apply as well. So the idea that any chemotherapy that has an atom that can bind to metal could be in principle used for doing these things.
Okay. And then Ben would like to know, would combining cytotoxic ruthenium complexes and cytotoxic ligands benefit the effectivity?
Yeah, this has been proposed in the literature. People have never really measured the actual benefits, or the really added value of the synergy, let's say. But we are working on this in my group. There is one PhD student working on this particular topic, and this is exactly the topic of my Vichy grant, which started last year. So we are trying to generate synergies between the ligand toxicity and the metal-based toxicity. So that is a very good good question. We are working on this, basically.
And lastly, have you also tried other caging groups than ruthenium?
Yes, there are. Ruthenium is the most interesting because it absorbs the furthest in the visible region, let's say, closest to the red and infrared region. However, people have used cobalt, people have used rhodium, and I would say the biggest competitor is platinum 4, so platinum 4 plus. There are light activated platinum 4 plus molecules, but still the problem is that it absorbs mostly in the blue. So ruthenium is still the best of all the metals I know because it absorbs in the green or even in the red and even in the near infrared. Now there are molecules that absorb in the near infrared. So that's really a strong advantage for ruthenium.
Okay, thank you very much. I enjoyed the talk. I would like now to point everyone to back to the main stage where the PhD quiz will take place. And I would say a big applause for Sylvestre, but I don't think he'll hear it.
Thank you very much for the invitation and for the questions.
Yes, thank you very much.
Enjoy the day. Bye-bye.